Okay, Assalamualaikum warahmatullahi wabarakatuh and very good morning. Okay, so today we will have a presentation for FIAT International Postgraduate Symposium 05 in conjunction with Undergraduate Symposium 01. Okay, here we have a physical presentation session 1. We will have a 7 presenter. So firstly, I want to remind you that you will have only 10 minutes to present and 5 minutes for Q&A. Okay. For first 8 minutes, we will remind you to conclude your presentation, and then after 10 minutes, you have to stop, and then we will have a Q&A session. Okay. All right. Without further ado, we would like to call for the first presenter, that is Miss Fadzlin Qistina Binti Fauzan, with the title Regeneration of Haploid Calluses from Ovary Culture of Cucumis Melo and its activity towards hormonal combination. Please come.
Hormonal Combination. So this is the content, right? So for the first part, I will start with introduction. Okay, so basically, the gynogenesis ovary culture is one of the ways of how we can obtain the gynogenesis, which we use to obtain the homozygous line instead of the anteculture and also the parthenocarpy embryo. So basically, the initial part, the gynogenesis, conducted in barley crop, and after it became a success, then the started to use it for the other crop as well. Agriculture usually is quite easy, but actually it is okay. It is okay. Okay. It's very dependent on genotype, the developmental stage, and also the several factors such as the media composition and so on. Okay. Okay, for next part, the this plant material that we use to study the ovary culture is the Glamour Japanese melon of C. Melo of Reticulatus varians. And it's actually they consist of both male and female parts. So, and the female part basically is studied for the parthenogenesis, which is the parthenocarpy embryo production, instead of being well studied for ovary culture. So, even if it is less favored by researchers, but it is still a very useful tool, especially when the anteculture and parthenogenesis is not applicable. Okay, so basically, what is the importance of the C. Melo ovary culture? So firstly, the ovary culture can be used to regenerate the haploid line of C. Melo plant. Secondly, the haploid line that is generated from the ovary can be used as the starting material for the dihaploid later on. And thirdly, here, it can be used as substitute of anteculture in the condition where the androgenesis is not applicable, such as when the albinism during the in vitro or regeneration is not successful, and so on. Okay, so this is the female flower of C. Melo. Okay, So I mentioned before about the dihaploid and also the double haploid. So basically, what is the double haploid? Double haploid is produced from haploid. So basically, we first produce haploid from in vitro culture, and then from the artificial doubling by using chemical, we can get the dihaploid. And from here, dihaploid will contain two similar homologs in which the amount of the recombinant inside the dihaploid is comparable to a backcross, in which the quality is quite high and it is agreed by many researchers. So importance of the double haploid. Double haploid is very important because, as I mentioned before, it can be a useful technique to produce high quality generation of plant and it is comparable to F1 hybrid. Secondly, the dihaploid will have fewer genotypic class. I will explain this later. And thirdly, double haploid population will have smaller population, so it is easier to observe all of the resultant. And fourthly, this is the most important part. At certain condition, the recessive allele in the plant can emerge because, as you know, that only dominant usually emerge because it is dominant. But for recessive allele, it can only emerge when both of the allele are homozygous, in which both are recessive. So that is when it can emerge from the allele to show its quality. Okay, so this is the segregation pattern of dihaploid and also F2 population. As you can see here, dihaploid population is quite small, so it is easier to observe to observe and validate the quality of each resultant. Okay, so next I will move to the methodology part. In methodology, firstly we start with collecting the flowers, and then we surface sterilize the flowers, and then we slice the ovary into several parts and culture onto the prepared media with the hormonal combination. And as you can see here is the experimental design in which the hormone used for the callus induction and maturation and followed by embryonic callus initiation. Okay, so we move to the result and discussion. So basically in the first part, the callus induction and also the maturation, actually this first part is the study that we follow based on the protocol by a study that has been conducted on cucumber species. And then in that study, the ovary culture is supplemented with this hormone, and we observe similar result when we compare to the study in which we successfully induce the ovary-derived callus. Okay, so on the opposite side, we also conduct the ovary culture on zero hormone media, and it shows no callus regeneration at all. And from this study, we can say that the callus regeneration depends greatly on hormonal presence. So this is the figure of where we extract the ovary into the culture. And next is the embryonic callus initiation. In this part, we try to initiate the embryonic callus or shoot. So here we study the effect of BAP at different concentration in constant addition of NAA. And from here, we raise the cytokinin level at higher ratio compared to the auxin because we aim to produce either embryo or shoot. So embryonic-like structure is important and because it can help to initiate the new plantlet. And here we can observe that 2B media containing 1.0 BAP and 0.05 NAA display the best response in which it gives the best formation of embryonic-like structure. So we can be sure here that the BAP present, the optimum level of BAP present with AAA, and AA give better formation of ELS compared to the NAA alone. Okay, so this is the result. This is the 2A media containing only NAA. This is the 2B media containing 0.05 NAA and 1.0 BAP. Okay, and this is the 2C media. So this is the evaluation. And this is the percentages of the ELS formation. You can see here that 2B produce the highest percentage of embryonic-like structure. So this is the quotation from a paper. The presence of both auxin and cytokinin at intermediate ratio will aid the process of callus initiation in plant. So as for the conclusion, this study conclude that the presence of both auxin and cytokinin always display the best callus regeneration compared to the application of either one of those hormone alone. And the ratio should be optimized because it can influence the regeneration rate of callus. So that's all from me. Thank you.
Thank you, Ms. Fadzlin, Qistina, for the next presentation. Okay, we have a first question from Dr. Nurafizah.
Okay, Ms. Fadzlin. Okay, actually, what is the most significant finding from your study just now?
My finding.
Yeah, the most significant finding.
The finding is that I found out that the callus regeneration give the best result when both auxin and cytokinin is combined at the intermediate ratio. If I only use the auxin alone, it will not give a good regeneration.
Okay, so can I know that you already get the callus right? So you said that the callus have embryo-like structure. So how you confirm that callus have an embryo-like structure? What you did to confirm that?
Okay, so basically, for this part, at first, the observation, I observed that the conformation of the callus is different from the first stage until to stage now. For the conformation, for what I did, but I did not state here, I did the chromosome counting actually. But by using callus, it's quite hard to get a clear chromosome, clear display of chromosome because of the debris from the callus. It caused like a very messy structure, so it's hard to count one by one. So for now, the future analysis that I will do is the cytogenetic method, such as the flow cytometry.
Okay, thank you, Ms. Qistina. Okay, now we go to the second presenter, that is Sir Muhammad Akmal bin Muhammad Zawawi. Please welcome. Okay, sorry. His title is about Internet of Things IoT technology to monitor agri-environmental of banana soil. Okay. Okay, because we have some issues, so we go to the next presenter, that is Miss Wan Nur Syafikah binti Wan Abdullah, with her presentation, Myorganic Adoption Framework Among Vegetable Farmers in Peninsular Malaysia. Please welcome.
Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. A very good morning to everyone. So my name is Wan Nur Syafikah binti Abdullah, and my supervisor is TS Teguh Halimatul Saadia. The title for my study is Myorganic Adoption Framework Among Vegetable Farmer in Peninsular Malaysia. So this study will be cover 5 parts. Okay, I will proceed to the introduction. Alright, Asia accounted for nearly 80% of all vegetable production worldwide, with estimate 898 million ton produced in 2020. And then according to DOA, the agriculture industry in Malaysia rapidly growing year by year, including the vegetable industry. And then vegetable crop value added to Malaysia gross GDP was predicted to increase from 12.15 billion ringgit in 2020 to 13.32 billion ringgit in 2021. So for my study, I will focus about organic farming. Organic farming is used to define agriculture which aim to minimize human impact on the environment while maintaining the natural functionality of the agricultural system. So for your information, there are 2 types of farming, which are conventional and organic farming. Compared to conventional farming, organic farming is the method that using natural resources such as organic fertilizer and organic pesticide. These are natural substances which do not damage the environment. Alright. In Malaysia, there is Malaysia Organic Certification, which we call Myorganic. So this scheme was launched in 2002 with the introduction of Organic Malaysia. And then in 2030, the government has changed the Organic Malaysia to SOM. And then in March 2050, the SOM was replaced by the new Myorganic logo, and it is now the sole national organic food logo. Alright, this graph shows the total number of farmers in agriculture industry in Malaysia that receive organic certification. So from this graph shows that the total number of farmers that receive certification is very critical, since the maximum number is 121 only. Okay, the problem statement for this study, the first issue is the total of vegetable farmer in Malaysia is about around 43,000. However, only 54 vegetable farmer that receives Myorganic certification. This number of farmers taking Myorganic certification are still low due to farmer mostly apply conventional method. And then this is the graph shows the all state in Malaysia that receive vegetable farmer, which only 54 of total vegetable farmers. And then the second issue is adoption of organic farming practice is low, because not all farmers are willing to adopt the organic farming practice due to not able to fulfill the commitment for organic practices in Malaysia. Alright, the third issue is only a few studies have discussed the impact of extension service on technology adoption. So this study will put the extension service as the additional variable. So extension service play a significant role in explaining the practice and technology adoption. Alright, for this study there are 5 objectives. The first is to determine the level of dependent variable, independent variable, and mediative variable. And then the second objective is to identify the relationship between independent variable and mediative variable among vegetable farmer. And then the third objective is to identify the relationship between facilitating condition and behavior intention. The fourth objective is to determine the influence of IV and mediative variable. And then the last objective is to determine the mediative effect of behavior intention on IV among vegetable farmer in Malaysia. Alright, the research question is based on the objective. Okay, in my study, there are 7 hypotheses. So this is the significance of study. For the new finding, extension service as additional construct variable will offer new finding on technology adoption theory, since this variable is still limited on research. And then for the society, a better understanding of organic farming practice will increase the number of farmers receive my organic certification. Then for the academia, literature based on this study will be used as reference for university and academia. And then for the government, this will promote the involvement of farmers, especially the vegetable farmer. And then for the environment, will promote the sustainable agriculture. All right, I will proceed to the literature review. Okay, for the adoption, most of the farmers are poorly adopt agriculture technology. This is based on the previous study. And then this study will using UTAU theory. And then this UTAU is the best theory to explain the technology and practices adoption. Right, this is the diagram for the UTAU theory. And then for the performance expectancy, from the previous study, performance expectancy, effort expectancy, social influence, facilitating condition, and behavior intention have significant on adoption in technology and practices. And then the second theory for my study is I will using TBB. This TBB have 3 elements, and this study I will using only attitude element because the rest of the element is the similar with the UTAU theory. Alright, this is the diagram for the TBB. And from the previous study, attitude has significant on the farmer to adopt organic farming. Alright, and this is the extension service. This is an additional construct variable. This is from the previous studies, extension service has significant relationship between farmer to adopt organic farming practices. All right, for the methodology, this is the research framework. And for the independent variable, which is attitude, social influence, performance expectancy, effort expectancy, extension service, which is additional construct, and facility condition. And this study also have mediator variable, which is behavior intention, and dependent variable, organic farming adoption. All right, for the research method, this study will be using quantitative to test the relationship among variables. And then for the data collection, I will be using primary, self-administered questionnaire, and structured interview. Then for the study population and sample size, the number of vegetable farmer is 43,086 according to DOA, and only 384 respondents will be using according to the Krige and Morgan model. And then for the sampling technique, I will be using cluster sampling. And then this selection will be according to the list number of vegetable farmer from DOA in a 4 zone, which are north, south, middle, and east in Peninsular Malaysia. All right, this is the population and sample size. So this 4 state is based on the highest number of vegetable farmer in Peninsular Malaysia. Okay, for the data analysis, I will be using descriptive statistic based on the objective 1, and then for the
Okay,
measurement
sorry, no Syafikah, you have 1 minute left.
Okay, next, the measurement model I will be using for the data analysis according to objective 2 and 3. And then for the objective 4, I will be using structural model. And then for objective 5, I will be using mediative model. Okay, for the expected outcome and for the conclusion, from this study, the level of DV, IV, and mediative variable have high mean score. And then for the relationship between IV and mediative variable will be evaluated. And then the relationship between facilitating condition and behavior intention will be evaluated. Then for the fourth is the influence between IV and mediative variable will have a strong influence value. And lastly, for the expected outcome is the mediative effect of behavior intention on IV will be test. Right. That's all from me. Thank you.
Okay, thank you, Wano Syafikah, for the next presentation. So we go for Q&A session.
Good presentation, one, Dr. Syafikah. So my first question is that what is the acreage determination that been set up for your experiment? And then the second one, is there any market demands in terms of organic vegetable produce that be incurred together in your studies?
The first is about determination. Okay. For the determination of my study, this study I will using extension service as the additional construct, since this variable is limited on the previous research. So this variable is very important to adoption in organic farming. So extension service is important for this study. Okay, for the market demands, my organic certification is very low in Malaysia, since the management practice for organic is very complicated for the farmer to follow the organic scheme. So that's why the market for my organic certification is still low in Malaysia.
I have one question. Okay, actually Malaysia there are two types of land pattern use in Malaysia, which is intensive and extensive farming. So which one of the farming methods that most likely suitable to apply my organic certification?
Right. For the extensive farming, this is for my opinion, extensive farming is mostly using organic method. So for my opinion, extensive farming is most suitable for the organic farming practices.
Okay, thank you, Wano Syafikah.
Thank you.
Okay, we go to the next presenter. So I hope that Muhammad Akmal is ready. Okay, please come Muhammad Akmal Muhammad Zawawi with the title Internet of Things, Technology to Monitor Agri-Environmental of Banana Soil. Please welcome.
Assalamualaikum and good morning to all. Sorry for the technical issue just now. I'm Muhammad Akmal Muhammad Zawawi. I would like to present a part of my objective, my ongoing project entitled Internet of Things IoT Technology to Monitor Agri-Environmental of Banana and Soy. So my project mainly supervised by Dr. Marina and co-supervisor Dr. Laila and Dr. Nurul Shaza from UITM Shah Alam. So IoT, as we know about the agriculture is the remains as important sector in our economy, contributing to GDP Malaysia. So why we need to implement IoT in agriculture industry? To improve agricultural productivity, to monitor the agri-environmental attributes such as soil moisture, air humidity, soil or another surrounding of the temperature. So in our current study, we implement or we focus on the banana, or specifically focus on the Fusarium wilt disease banana. So throughout the presentation, I will use abbreviation FWB, referring to Fusarium wilt disease banana. So the problem statements, as we know that FWB is the remain or the major constraint in banana plantation. To date, there is no effective cure or treatment to prevent this disease. That's how we come out with the IoT technology. So far, there is no systematic monitoring and detection for the FWB disease transmission has been reported in the literature. And then the second one is about the traditional agricultural practices that currently implementing in banana plantation. There is no effective detection or early detection regards to FWB disease development. And the third one is about the less clear relationship about the agro-environmental factors either directly or indirectly affect the FWB disease. According to the literature, there are two contradictory opinions. For example, some literature or scholars found that low pH level associated to the FWB disease, but other scholars found that high pH level associated to FWB disease. So that's why we come out with the time series system by implementing IoT technology. So for the research objective, for today's presentation, we just focus to introduce or to highlight the development of the affordable IoT system to study the association of the real-time data of agro-environmental and FWB disease, or shortly known as forecasting system. So for the research methodology, today's presentation only cover the soil moisture sensor, humidity and temperature sensor, or also known as DHT22, with the microcontroller ESP8266 Wi-Fi module. So the software implemented in this study, Arduino IDE, the software that we use to code, to program the ESP8266, and the ThinSpeed for the cloud and visualization. So the result and discussion to test, as I said earlier, today's presentation, just to highlight the functionality of the sensor. So the soil sensor was tested to produce different level of soil moisture, air humidity, and air temperature. As you can see from the ThinSpeed on the picture on the left slide, the low air temperature produce high humidity and high soil moisture. So it means that our system, our coding, can read the sensor and produce the result and can visualize on the ThinSpeed. Another one, we use ThinSpeed because we can directly analyze the result using MATLAB. So we can create the graph as we can see on the picture. Also, we compute the descriptive statistic directly from the MATLAB. So current work, we are currently calibrate the sensor because we cannot rely on the factory calibration because sometimes the reading is not stable and then it's not accurate. Since this project involves mathematical modeling, we want to predict or we want to develop mathematical modeling progression for FWB. So that's why we highly concerned about the reliability and accuracy of the data that we collected. So that's why on the current stage, we are developing the calibration methods. After that, we implemented for the time series to get the data, and then we compute the mathematical modeling. For the mathematical modeling, we're using the survival analysis that is rarely used in the agriculture industry, especially for the plant pathological research that commonly used. Survival analysis only common use in the medical field. So we want to introduce in plant pathological research because it rarely used by the researchers. So conclusion in this presentation, the design, as I said earlier, the design and the deployment of the real-time banana system for the smart agriculture had been presented. And then this sensor capable to measure the soil properties, for example, soil moisture, air humidity, and air temperature. And as I said earlier, our next current work is to calibrate the sensor and implement this technology in the greenhouse so that it can collect the time series data before we develop mathematical modeling. And that's all from me. Thank you.
Thank you, Akmal. Very good presentation. Okay, my question is that you mentioned on the pH, okay, the variation in the pH that, determine the level of a infestation of FWB in the soils. Have you done the testing, the calibration with the pH sensors? And what kind of pH sensor that you're going to use for your studies?
Okay, for the pH sensor, we using the integrated soil moisture sensor, which could measure the soil pH, soil electrical conductivity, soil nitrogen, phosphorus, and potassium. So this is the second part because the second part of the system, I think this one.
I see.
This one is the soil that measures the soil pH, soil
Altogether.
nitrogen, electrical
Altogether.
conductivity.
Have you done the latency of the data that been transmitted from your? Are you using Wi-Fi or using the wire? Everything.
Using Wi-Fi.
Wi-Fi, using the wire.
No, a bit.
I think you need to be considered on the wire altogether in case the Wi-Fi been disconnected from the connection.
I see. Because this system, actually, I forgot to highlight that we also use backup data. I mean, like we already code the coding to, it means that we use the SD card. So whenever the internet connection corrupt, the data will be uploaded to SD card.
I see. So it will be, if the Wi-Fi means the data will be continue
Yes.
recording of this your data.
Yes.
Okay,
Yes.
Understand. So, is there any preliminary data on the findings? Is there any correlations of your data with the FWB or any literature review that have been finding from your studies?
Okay, the development of IoT is the second my objective. The first objective, we conducted the preliminary to study the characteristic of the soil that are associated to the FWB disease and the soil properties. So the findings, like I said, is very contradictory with the literatures. My finding is low pH level and high moisture content associated to the Fusarium wilt disease. But in the literature, like I said earlier, there are two opinions. Some say low pH level and some say high pH level associated to the FWB disease. So my first finding, what I can say is parallel with the literature. But in terms of the IoT itself to monitor, I mean to read the soil properties, is still not testing because we are at the stage of the calibration method. After done the calibration method, we tested to the soil banana and we insert the pathogen because we want to study FWB progression. Either FWB change the soil properties or soil properties change the development of the FWB. So that's why I said in the problem statement, we know clear relationship, either directly or indirectly affected FWB. So we hope that time series system could reduce the gap between the relationship.
Okay, last one. Can you explain on your experimental designs, your design for your studies, and then with the time series one, so that with your sensors, so that it makes sense in terms of your studies?
Okay, I highlight for the IoT system part. Okay, I was tested in a greenhouse. It means that 20 pot banana plants. It means that 10 for the control and 10 will be inoculation pathogen. So 20 replicates of this system will be implemented in the greenhouse for 2 months. Because why we selected for 2 months? Because we are using the soil moisture sensor that exposed to the corrosive. Because if we want the what we call it, the high advanced soil moisture, of course we involve high more cost. So for this preliminary study, we are using a very cheaper soil moisture sensor implemented in the greenhouse for 2 months, and then the data will be collected hourly, and then using the average of daily we will do for data analysis. For the first part, we want to determine the threshold value. That's why we are using the survivor analysis. Survivor analysis means that at the what day the symptoms of FWB expressed at the physically. So at that day, we stopped the experiment and we conducted the survival analysis, and then we can determine the threshold value, what value of the pH associated with the FWB, what value of pH, soil moisture, and other soil attributes that associated to FWB. So that's the stage or the experimental design for IoT in the greenhouse. Okay, that's why because in the greenhouse study, we collected the data from day 0. I mean like before the pathogen inserted in the house, in the soil, and then we monitor the progression, okay, until the symptoms of the FWB expressed on the banana tree, and what we call it, we extend the experiment until the banana tree collapse. Okay, so to okay to okay to identify either pH or FWB affected to one another, we are actually with my supervisor, we expert in mathematical and statistical. We are trying to look at all the perspective of the data. Maybe at the first we try to run soil affected to FWB and turn over FWB affected the soil. So we are looking for the statistical analysis on that part, not for that one. I mean, like we try to look at with the data. Okay, for currently in the literature and my findings, soil attributes, what we call it, affected or contribute to the FWB development. So we need to, what we call it, but what I said is a very unclear relationship. Even in the literature also, they still debated about this one. So that's why we try to come up with the time series. Yes, until we try the system. For the soil. Okay, the soil, what we are discussing before, not sterilized. I mean, we because the reason why we don't sterilize, because I already said, because with the lab assistant, because yes. Yeah. Yeah, actually, we proposed to sterilize, but the lab assistant said it's too much soil to need to sterilize. Uh, okay, okay, okay. Okay, okay, thank you for consideration. I don't. Okay, sure. Okay, thank you. Thank you for suggestion.
Okay, thank you. Thank you for the next presentation and also the interesting research from Mr. Muhammad Akmal. And thank you also to the Dr. Raimi and Dr. Nufaiz for giving the suggestion to him. Okay, now we go to the next presenter, that is Miss Nur Salsabila Binti Shafiee, with her title Isolation and Identification of Plant Growth Promoting Fungus of Phalaenopsis Amabilis Blume Orchid.
Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. My name is Salsabila Binti Shafiee. Today I will presenting about my first objective, which is the isolation and identification of plant growth promoting fungus or Phalaenopsis Amabilis Blume Orchid. Okay. First, the background of the study, which is the Phalaenopsis Amabilis Blume Orchid. As we know, Phalaenopsis is one of the most Phalaenopsis is known as one of the epiphytic orchid that contain a large variety of the endophytic fungus that we're focusing today. And the endophyte fungus itself, as we know now, nowadays started to attract the attention as in finding the plant's growth promoting strain, because they are useful in enhancing the crops yield and also the quality of the crops. And next is for my problem statement. As my problem statement is focusing on my third objective, which is acclimatization. So the problem that we will face in the future, which is which we will face in the future, is that there are high mortality level after we acclimatize the plantlet from the tissue culture into the greenhouse, and also the greenhouse have a very low in relative humidity and also have a high level compared to the in vitro conditions, as will become problems to the plantlets and also the plantlets is unable to develop the resistance against the biotics and also abiotic stressor compared to in vitro control conditions. Next is for my objective. My focusing here is my objective one, which is to isolate and also identify the fungi from the three different parts of the Phalaenopsis Amabilis Blume Orchid, which is leaf, stem, and also the roots, by using morphological observation and also molecular analysis. For my literature, overall literature, which is consisting of these three parts, which is for the Phalaenopsis. Actually, Phalaenopsis have a root, stem, and also leaf, because they have, as I mentioned before, they are one of the epiphytic orchids, and the endophytic fungi itself can improve the growth and reduce the effects to the both biotic and abiotic stressor. And next we have for the direct observations. So we are using I will present the result soon, which is we examine the endophytic fungus structure using the microscope. Next, my method, which is I focusing for my two method, isolation and so identification, for my objective one. For the isolations, we cut the three different parts of the Phalaenopsis, which is from the leaf, stem, and also the roots. We will cut them into 2 to 4 centimeters, 40 meters, and then we will treat them with a surface sterilization using 70% of ethanol, 2.5 sodium hypochlorite, and also we will rinse them with this dry distilled water for the treatment time. And all of this method will be done in the dry conditions. Next, for the isolation part, for the inoculation, we use a leaf imprinting method on the PDA media, and we incubate at the room temperature for 7 to 10 days for monitoring. And next, for the maintenance, we use the continuous subculturing within 7 to 10 days to obtain the pure culture. And also we use the slang culture, and we keep them at the 7 to 8 degrees Celsius as a permanent culture. And also for the glass raw stock, we keep the fungi in the glass raw stock for long-term preservation. Next is the morphological. We are observed using using microscopic observation and also microscopic observations, which is we did the slide culture and then we incubate at the room temperature after 7 to 10 days of the incubation, we will observe the slide under the camera microscope, light microscope. For the result and the discussion, for the isolation, we gained around 15 isolated fungi here based on and we group them into 8 group, which is the group divided into 8 groups based on their physiological appearance like color, surface texture, shape, and also the growth rate. Next, for the physiological characterization, we will monitor within 7 to 10 days of monitoring of the fungi. You can see from the group 1, there are the same thing, which is the color is white and also dark brown on the reverse, and the texture is a bit cottony. Also, the shape was round, which is regular, and the growth rate in the fast growth. And this is quite different from this group 1. This group 2, they have a color white and also browny, but the shape is quite different. But I grouped them in one group because of the color and also the texture, and the growth rate is the same, which is a slow growth. And that's for the group 3. We have quite different from before, which is they have a white and also white creamy on the reverse, and the texture, the chalky texture, and the shape is regular, and their growth rate is very fast within 7 to 10 days. And for the group 4, we can see the color is quite different, which is they have a dark grayish on the surface and also have a gray margin, and they have a reddish on the reverse. Next is the texture. It's come off cottony, also fluffy a bit. So the shapes looks like irregular, and the growth rate is a fast growth. Next, for the group 5, we can see it's very different because they have a dark green color on the surface, white margin, and also grey-green on the reverse. The texture is a velvety, and the shape is irregular, and also the growth rate is a slow growth. Compared to group 8, we can see the colors is dark grayish and also have a grey-beige margin and dark grey on the reverse, and have a hairy and fluffy shapes. The texture and the shape is regular, and also the growth rate is fast growth. For these two groups, groups 7 and also group 6, we can see the color is white and also orange creamy on the reverse, and the groups 8, we can see white and browny white on the reverse, but the growth rate is a slow growth. Next is for the anatomical characterization. You can see after we monitor under the light microscope, we can see that for this group 1 and group 2, they have a hyphae with a septate, but in the group 2, we can see they have a formation of some of microconidia, and also group 1 where they have a swelling hyphae. Next, for the group 3 and the group 4, they have some network of the hyphae, and the hyphae have a non-septic from the group 3. For the group 4, they have a intercalary hyphae swelling, and also there is hyphae with the septic. Okay, for the group 5 and the group 6, we can see the group 5 have a conidia spore and also have a non-septic hyphae, and it's a clump of the conidia spore. And for the group 6, we can see there is a non-septic hyphae, and there is some pseudo hyphae at the hyphae. Okay, for the group 7 and the group 8, there is a septic hyphae, and group 8 have a swelling hyphae. And for the conclusions, I conclude that some of the fungi, mainly from the Ascomycota phylum, is basically they have a septate hyphae, and with the simple septal spore, and from the morphological identifications, few of the isolates show like the same characterization with the same and identify endophytic fungi, but we still cannot finalize the result until we did the molecular identification. This is my reference, and thank you.
Okay, thank you. For the next presentation, Nur Salsabila.
Okay, I have two questions. The first question, where do you collect the plant sample, means like the orchid? And number two, what type of tool or equipments that you use in your study, especially on for microscopic parameter, for example, like texture, color?
The plant sample I collect from the Phalaenopsis, purple color of the Phalaenopsis. I take 3 parts of them, which is the leaf, roots, and also the stem. And then for the microscopic, I monitor the texture, color. Location of the Phalaenopsis from nursery in the UMK. Okay, for the microscopic, I monitor the texture of the fungi after I contain the pure culture. I monitor the texture, the colors, and also the growth rate, because some of the endophytic fungi actually
Is it all based on your observation, or you use any tool or equipment?
for the macroscopic, I only use just by naked eyes by seeing, and also did some research on the.
So, is there any literature that describe the criteria for each of the parameter, for example, like color? Is it any criteria?
Okay, for the endophytic fungi that I focusing, some literature said like the colors and also the growth rate show like slow growth for the growth rate, but as we can see, my results, some have a slow growth and some have a fast growth. So from there, I can like divide them like some.
How are you going to exclude because you want to select those fungus that are actually promoting the growth of a specific orchids, right? That is endophytic inside the orchid. But how would you exclude those fungus that are the normal, usual microbiome inside the soil, or it may not be related to the orchid itself, but how are you going to select the one that is specific?
Before I inoculate them in the PDA media, I did the surface sterilization.
Yeah, because I already tried that too, but and then we sequence, but with my previous experience, we're still sequencing the normal microbiomes that are existing in soil, even though we surface sterilize them. So you need to find ways on how you're going to capture the right one inside. So you need to have some sort of expectation to the fungus that are playing role. You need to find ways and method to detect the right one that helping or promoting growth of that orchids. How are you going to capture that? So if in my case, I'm trying to find bacteria that are causing disease to the pineapple, and we're trying to isolate the bacteria that causing the disease, right? So mine will be easier. I just need to take the bacteria and put it in a pineapple and see whether or not it caused the same disease as I expected. So I guess you also need to find ways on how you want to find link between the right fungus that helps the growth or promoting fruiting or flowering of the orchids or something. What is their role, the fungus role? So that can be used in order to capture the right roles of the fungus and the one that you are isolating, so that you're not isolating just the normal microbiome. So that is my concern. you know what microbiome, right? The normal, usual, yeah.
I will discuss later. Thank you, Doctor.
Okay, thank you, Miss Nur Salsabila, for the next presentation. Okay, next we will go to the next presenter. That is Miss Nur Shafira Shuhada Binti Fazil, with her title Nutritional Composition and Antioxidant Activities of Pleurotus pulmonarius, Growth with Different Substrates. Please welcome.
Okay, Assalamualaikum warahmatullahi wabarakatuh, and good morning to all the lecturers and friends. So today I will present about nutritional composition and antioxidant activity of mushroom, specifically Pleurotus pulmonaris stem waste with different substrate, which is sawdust and pineapple waste. So for the introduction, mushrooms, specifically the Pleurotus genus, consists of 40 species that have grown widely all over the country. Mushroom also have been consumed due to the unique flavor, rich in nutrient and other secondary metabolites such as polysaccharide that have medicinal properties. It also have been chosen by multiple mushroom producers due to the easier cultivation method and lower cost of production. Besides, oyster mushroom also has known as the world's third highest record of cultivated mushroom after the shiitake and button mushroom. In our country, Malaysia also, mushroom are categorized as one of high value crop, HVC, such as kelulut honey, pineapple and durian due to high market demand, shorter harvest period and profitable output. So this anatomy of the mushroom. So for this study, we have divided the sample to 3 parts, which is cap, stem, and the stem waste. The stem waste is the bottom part of the mushroom that usually have been discarded by mushroom growers before sell it to the retailers. For the problem statement, the rising number of mushroom production, specifically in our country, has produced a large amount of waste, which is a stem waste that I have mentioned before. And to preserve our environment quality, we have to recycle the stem waste in a good way. So it also has been pointed out that there is still lack of a study present in the stem waste from the mushroom farming industry, specifically the stem waste. So our objective is to evaluate the nutritional composition and also the antioxidant activity that present in cap, stem, and stem waste cultivated with 2 different substrates. For materials and method, the sample preparation, the mushroom block have been purchased from agro farm in Pontian, Johor, and kept in the mushroom house in our university. The sample also were collected, cleaned and separate between cap, stem, and stem waste, and dried in oven for 2 days, ground and kept in airtight container until further analysis. So here is the table show that percentage of ingredient that used in the substrate. So for the pineapple waste substrate, the waste part that have been used is the leaf of the pineapple, and also the substrate is the mixture of the pineapple waste and the sawdust, as shown in the table. For the sample extraction, we have been macerate the sample with 80% ethanol for 24 hours at room temperature. Then the sample was filtered, evaporate, freeze dry, weigh, and store at negative 80 until further analysis. So for proximate analysis, the fresh sample have been used in this study. The 6 elements that have been tested is moisture content, ash, fat content, where petroleum ether is used to extract the fat, and the defectant sample from the fat have been introduced to the crude fiber analysis using the automated system FiberTech. and protein content using Jadal method that basically involve three basic principles, which is digestion, distillation, and titration against acid to obtain the total nitrogen content. And the last one, the carbohydrate content can be obtained by subtracting all the of the element by 100. Next, for the glucan composition, we also have measured the alpha and beta glucan. So the beta glucan content is calculated by subtracting the alpha glucan from the total glucan content. This test is basically involve the solubilization, hydrolysis, and the enzymatic hydrolysis using an enzyme, which in the last experiment, we got the color changes that can be measured using spectrophotometer. Next, for the antioxidant activity, also involve the color changes in the experiment, and basically it involved the radical compound react with the antioxidant compound, which is our sample. For the result and discussion, we have the table is the superscript in the table show the different statistical significant value. The PS is referred to the pineapple substrate, and SS refer to sawdust substrate. And also in the third row, you can see that we use the letter C, S, and SW to refer the three parts of our sample. So we can see that PSC had significantly higher moisture compared to others, and PSC and SSC, which is the cap part for the both substrate, was significantly higher in protein. And for the third one, stem waste and the stem is significantly higher in carbohydrate compared to the other part. However, there is no significant difference in ash, fat, and fiber composition between all the parts. We also have compared our result with previous study that have been done by Chang and Srikram and Supavarnish. We can see that most of all the elements were in range because of the sample that we have used, and the previous study is a fresh sample and quite similar condition, growing condition, and also the species. However, in carbohydrate, our value is quite high due to our sample that have been separated into three parts, and the previous study have been used the whole mushroom. For the glucan composition, there is no significant difference in alpha glucan content between all parts in both substrate. However, the stem and stem waste have significantly higher amount in beta glucan than the cap. Our result also have quite similar to previous study that report amount beta glucan in the mushroom in separate part are between 32.5 to 50%. Our data also is in agreement with Sari et al., who found that beta-glucan content is abundant in their type compared to the cap. This is because the stem is naturally conduct a wide range of process to enable a good growth of the mushroom. For the antioxidant activity, we can see that the IC50 value in the cap is higher than the stem and stem waste, with the range 231 to 272. And the IC50 value also show that our sample had higher antioxidant activity compared to the previous study that have been done by Arbayah and Umil Kasum, who found that IC50 value is...
You have 1 minute left.
Okay. From 2.75 into 12 milligram per mil. And okay, for the conclusion, it can be said that the different substrates that have been used in this study have no significant effect on the nutrient and antioxidant activity. The stem waste also have showed similar nutrient composition compared to other parts for most of the element. And the stem waste show a higher beta-glucan content compared to the cap and higher antioxidant compared to the stem. And the stem waste has its own potential, and the active compound can be explored, which may contribute to the pharmaceutical and cosmeceutical industry in the future. That's all for me. Thank you.
Okay, thank you very much, Ms. Nur Shafira Shuhada. Next presentation. So, okay, Dr. Faiz have a question.
So, thank you. Good presentation. What is the future application from your result finding of these studies? Your future application? Yes, correct.
Future application, we want to apply the compound actually in the, I think in the cosmetic product. I think okay. Yeah, in the future we plan actually. Okay. We aim to put the active compound in the product, especially in the cosmetic or in food maybe. We will do some the test upon the something about what we call something about the cosmetic like anti-aging, antioxidant benefit, and we'll set. Yeah, because of the we how to explain to the consumer or what? Convince. Okay. Okay, maybe we can. Oh my God. Actually, we can convince on the benefit, and we can show the process during extracting the compound. Where we clean and sterilize, or okay, we cannot sterilize the waste, so that the consumer will confident that okay, this is a good one. And also, I have heard that many waste of other than mushroom also have been used in and applied in the cosmetic, right? Such as in our friend the watermelon waste that applied in the lotion. So I think the consumer may accept.
Actually, by calling it waste tu yang buat consumer tak suka. Actually, it can be called by other thing, by product, you know.
Okay, yeah.
So yeah, but my concern about your technical thing is that when you try to get the stem must be the one that is closest to the substrate, right? The one that you call as a waste. It must be the stem, because the mushroom is actually the fruiting body, right? And before it actually produces the fruiting body, it's just a mycelium inside the substrate. When it is at the right condition, then the fruiting body will appear. So the stem that you collect, that you call as a waste, usually are the one that is attached to the substrate, right? So when you actually do your analysis just now, how do you ensure that when you take the stem that you call as a waste, it is free from the substrate?
From the substrate.
You ensure that it is free from the substrate, because when you actually buy the mushroom, when it comes together with the end of the stalk, which is the end of the stem, it has a bit of the substrate left over. So that's my concern. So you need to really be sure that it is free from the substrate and it's only the stem remaining. Because if you're talking about the stem that are clean, those stems are actually consumed as a food. I think your research is important because you don't want to compete with food. You don't want to compete with food. So that's what you're supposed to answer Dr. Faiz just now. You don't want to compete with food. So you want to use things that are non-edible and it can be used for other products. So when you talk about that, the part of mushroom that is not edible is usually the one that with the substrate. Sebab itulah substrate tu memainkan peranan with your nutritional content tadi tu. So you need to be careful with that. Well, I mean, I'm not sure whether you need to be careful with that or not be careful with that. I mean, the substrate may be safe also to be used in a cosmetical product. So it actually makes sense why you want to test those substrate, different substrate, to get better better mushroom with a better nutritional content, because it comes together.
Question from me. Actually, how do you determine your substrate formulation? You follow from previous study or you formulate by
Substrate.
yourself? Yes.
Actually, we just use a different regularly substrate that have been used by growers. As we all know, the commercial substrate have been used is soda substrate. But we found that in Johor, they are commercially sell the pineapple waste substrate. So we just take it because we don't want to grow by ourselves to save the time. And then the pineapple have been used.
Okay, so this is your last semester here? You still have other experiment to do?
Yeah, there's still a little bit.
Okay. Okay, thank you, Shafira, and thank you to all the panels for the suggestion. So we go to the next presenter, that is Mr. Basiri Briston, with his title, Effect of Soil Factors on the Phytotoxicity and Persistence of Bis 2-ethyl hexyl Fertilate. Please welcome.
Physical. Yeah, I'm not used to. Yeah, I'm going. Good morning, ladies and gentlemen. My name is Basiri Briston. I'll be presenting part of my research work, which is titled The Effect of Soil Factors and the Phytotoxicity and Assistance of Bis 2-ethyl hexyl Fertilate. This compound it goes by different synonyms, either as bis-ethyl Fertilate, di-2-ethyl Fertilate, or dioctyl Fertilate. And it's one of the most common compounds that can be synthesized in the laboratory and also can be synthesized by microorganisms. Now, it accounts for more than 1/3 or 80% of the chocolate production in the EU and China. It is a cosmopolitan compound that is found distributed in different ecosystems, ranging from the soil, the air, the water. And it is used in a lot of products, ranging from medical equipment to building, to manufacturing, and personal healthcare products, though it's been reported to has a lot of toxic effects depending upon the concentration. Now, this particular compound, it has been reported to be synthesized by different life forms, ranging from bacteria to fungi to the algae, as well as higher plants. Now, amongst the bacteria, it's been produced by different species of bacillus, and also among the fungi, it's produced by Aspergillus species and the Coscocum nigrum. Now, large production of this particular compound is being found in the higher plants. It's been produced amongst 14 classes of 14 botanical classes, and amongst these we have plants like the Tura stromonium, the Alculina cordifolia, the aloe vera, Coibia, the Patinum stiriforus. Now, it's also been reported to demonstrate a lot of antibacterial activity against both gram-negative as well as gram-negative bacteria. It also has antiretroviral and anti-tumor activities. It also inhibits the growth of certain fungi, ranging from the Aspergillus parasiticus, Fusarium, and the Penicillium. Now, it's also reported to have antibacterial and larvicidal activity against the mosquito species known as the Ilex species, larvae. Now, it's also been reported to possess certain herbicidal activities. It's able to inhibit the germination of different species of plants, ranging from the Cucumis tobacum, the Cucumis sativus, the Brassica caensis, the Clona. Now, it has poor solubility, and because of that, it dissolves and gradually gets into the groundwater. Now, among different sources of this particular compound is found used abundantly in agriculture, whereby they use it for purpose of mulching. And in mulching, it releases some parts of this compound naturally into the environment. So this study is actually aimed at evaluating the persistence of this particular compound in the soil and its effects on the physicochemical and biological parameters if it is to be used as a natural herbicide. Now, this is the flowchart of our methodology. We now have the source of the samples. Seeds of E. chelona were collected from pested fields, paddy fields particularly, because E. chelona is a wheat of paddy. Now, this particular compound, it was purchased from Sigma company in Selangor. Now, soil samples were collected from the agro park. Then they were brought to the lab, dried at room temperature. Now the soil was subdivided into 2 batches. The first batch was to be sterilized, and the second batch was left unsterilized. The essence of the sterilization is to ensure microbial killing some common bacterial colonies that are found in the soil can be sterilized or can be killed using heat. So the soil was autoclaved at 121 degrees centigrade for 15 minutes, depending upon the cycle. Then after that, both the sterilized and the unsterilized soils were weighed 25 grams each and placed into petri dishes. Then these petri dishes were actually divided into different batches because the experiment is going to span for a period of 7 weeks. Now, the compound was prepared in different concentrations, ranging from 1.2 to 2.4 and then 4.8 kg AI per hectare. That's the application rate. Now, before the preparation, we now use DMSO as a solvent or as a diluent, and then we also use saponin to ensure mixability. Now, out of this, we now use 25 seeds of each of the compounds in the petri dishes. Then we apply the different application rates to evaluate the effect of this compound on the emergence of the seeds of the wheat over time. Now, the cycle for each of the experiment was actually 7 days. So 7 days, that is 1 week. So after every week, the experiment was terminated. We now put in a new set. But meanwhile, all these concentrations were applied. 6.5 ml of the compound was applied in each of the petri dishes right at the beginning of the experiment. So we have 7 batches that will cover 7 weeks. So, but the compounds were applied right at the beginning to see how it will persevere in the soil and to also look at the effect of microorganisms on that particular compound. Maybe if these compounds may have the potential of degrading it via natural process. Because the first batch, we've already deactivated the microorganisms via microbial killing. Now, after the seeds have germinated at every week, we took data on the percentage of emergence, also took data on the shoot length, as well as the root length. Then after the end of the experiment, at the end of 7 weeks, then pull the soils together, going by the different concentrations. And then we had to now analyze the physicochemical parameters. The physical parameters include the cell texture. Chemical parameters include the pH, the nitrogen content, the organic carbon content, the cation exchange capacity, and the microbial parameter that was evaluated was the colony forming unit. We did that, and all the standard procedures for.
Sorry, Mr. Brewston, you have 1 minute left.
Okay. Statistical analysis, we actually use ANOVA, and means that were significant were separated using a Toki test. Sorry. Now, figure 1, you can see we have sterilized and unsterilized soil. So we have the weeks. So if you look at week 1, week 2, week 3, the germination was actually impeded on the sterilized soil. And also, the other one is the unsterilized soil, you can now see. So at the end of 7 weeks, we were able to get a significant percentage of germination. Then on the sterilized soil also, the effects on the shoot length, you can also see at the end of from the 5 week to the 7 week, you can see substantial growth. That is to say, the compounds have been reduced. Similarly, the root length. Then these are the effects of the compound on the physicochemical parameters. You can see on the unsterilized soil, you can see the control is 5. So there was actually significant difference in the pH with the treatment. But if you look at the phosphorus and the potassium content, the phosphorus content, the potassium, it increases with the increase in concentration, likewise the CEC. Then the next table also shows the effect, the results of this compound on the unsterilized soil. Then our conclusion is that we've noticed that the presence of microorganisms actually in the non-sterilized soil help in the degradation of this particular compound over time. And also the effect of the compound, it has no significant effect to some extent on the physicochemical parameters, some of the physicochemical parameters. So based on that, we want to conclude that this particular compound, with a lot of consideration, it can be used as, or can be developed into a natural bioherbicide. Thank you very much.
Okay, thank you, Mr. Basiri Briston, for the next presentation.
Thank you. Very good presentation. Okay. As I say that this, the HAP may lower the, the toxicity. I mean, how long is basically the toxicity being reduced based on your finding? Is it, the faster is it a week, 2 days, or for the prolonged long time? How is the effect? How long you been done your studies?
Yeah, based on our experiment, for us to achieve, maybe for the emergence to be 100%, that is to say, the presence of the compound is no longer available in the soil. It starts panning from the 5th to 7 weeks. But there are other researches that stated that this compound gets degraded within the soil within 21 days. It's already wiped out soil.
Then, are you consider the, external factor or the weather factor, for example, like erosions, runoff, or water movement inside in the soil during your study in terms of the rate of the toxicity being lowered during your studies?
Yeah, this particular experiment was actually done in the laboratory. It was a Petri dish experiment. But also, we were able to collect the soil from the field studies we did, where all the climatic factors, the edaphic factors come into play via natural process. So we collected the soil and then discovered that actually the compound did not have much effect on the microbial flora and the physicochemical parameters. But what we were unable to cover in this area is look at the specific species of either bacteria or fungi that are responsible for the degradation. Okay.
Oh, are you specify the bacteria being, played a role in terms of the toxicity rates in the soil? Are you identifying?
That is what I've said. I said for us, we actually looked at the CFU only, the colony count. We just look at.
But you don't identify what kind of.
That is what I said. That is the limitation of this particular study. And that one can be suggested.
So how about the compound itself that attached with the soil? In terms of the HCP, is it take longer time because in terms of being the rates of the lower toxicity, are you done the study in terms of the interaction compound within the HCP with the soil particle itself?
The binding of the compound with the soil. Actually, that particular compound, it gradually like a little bit. It goes into the soil. It's released gradually, but going by it's also to some extent it's a little bit to some extent like volatile compound, but it mixes with this water gradually. But when it is with soil or organic matter, it binds tenaciously to the soil particles, and it's being reported by different authors as well.
Okay, thank you very much, Mr. Basir Briston, because we have a limited time now. We have a time constraint.
All right.
So thank you for your presentation.
All right.
Okay, so we go to the last presenter for this session. That is Miss Siti Mariam Salamah binti Abdul Rahman, with her title that is Effects of Different Substrate on the Quantification of Beta-glucan Content in Mushroom Species of Pleurotus ostreatus, Pleurotus florida, and also Schizophyllum commune. Please welcome.
Assalamualaikum and hello to the judges and audience. I'm Siti Mariam. I'm going to present my project regarding the effect of different substrates on the quantification of beta-glucan content in mushroom species of Pleurotus ostreatus, Pleurotus florida, and Schizophyllum commune. So these are the whole overview for this presentation. Mushroom industry has increased in the prominence due to its diverse applications toward human, where the wide range of the edible mushrooms in Malaysia has led to the growers to increase its productions. Most of the consumer, they consume mushroom because of the nutrition content apart of being tasty food. This is because edible mushrooms contain bioactive compounds that can benefit human health, such as beta-glucans, which they have function in cholesterol level, lower the cholesterol level in human bodies. Mushroom also are known for their therapeutic healing properties, which has established various benefit in traditional medicines and pharmaceutical applications. But beta-glucan can be found in mushroom where mainly comprised in the cell wall and primarily made up of beta-D-glucose. So for the problem statement, since different agricultural biomass selected as alternative substrate in mushroom cultivation, there might be also difference in the quality and the nutritional component in the mushroom fruit bodies. However, limited study or no data was found for the substrate can significantly affect the beta-glucans in the mushroom. So the objective is to observe the effect of different substrate on the beta-glucan content of 3 different mushroom species due to the different preference of mushroom species among the mushroom consumer. For the method of this study, it started with mushroom cultivations where 3 agricultural biomass used for the mushroom cultivation, which are sawdust, the commercial one, oil palm frond, and paddy straw. This substrate were cultivated by grey oyster mushroom, Pleurotus ostreatus, white oyster mushroom, Pleurotus florida, and split gill mushroom, Schizophyllum commune. And then the mushroom will be cultivated, and during the cultivation, humidity and the temperature were controlled. Then the mushroom were harvested for the grey oyster mushroom and white oyster mushroom. We harvested for 3 successive flushes, which are the first, second, and fourth flushes. While for the Schizophyllum commune, the split gill mushroom, we cultivated only for 2 successive flushes, which are first and second flushes, due to no formation of fruiting bodies. So we prepared it by drying oven method, which is using 50 degrees Celsius for 20 hours. And then the dried mushroom sample will proceed for beta-glucan content, which the analysis content of total glucan content and alpha glucan content. For total glucan content, it consists of solubilization and partial hydrolysis, where I digest a sample using sulfuric acid for 2 hours. And then for alpha glucan content, I do for the solubilization and hydrolysis and measurement of alpha-glucan. For that, for all the sample, I measure the absorbance and the color changes toward the samples. So for beta-glucan content, it is determined by deduction of alpha-glucan content from the total glucan which remains for the beta-glucan. From the findings, mushroom cultivated on oil palm substrate produce the highest total beta-glucan, which is in grey oyster mushroom, the highest one, followed by white oyster mushroom and split gill mushroom, compared to the other two, which is the sawdust and paddy straw. It also can be noted from the study that mushroom species, substrate element, environmental factor, and mycelium strain sources can impact the amount of beta-glucan in the mushroom fruiting bodies. It is also noted that there was definite difference in the nutrient in the substrate, which has contributed towards the difference in the beta-glucan content in the mushrooms. So oil palm frond is reported to have high sugar content, which can be the contributors towards the enriching nutrient, especially the beta-glucan in the mushrooms. So according to the Quattrosis et al. 2019, the nutrient component in the mushroom fruit bodies can be significantly increased by adding phenolic and antioxidant-rich materials to the cultivation substrates. And for the conclusions, it shows that substrate element could really impact the amount of beta-glucan content in mushroom fruit bodies due to the different nutrient content in the substrates. And it also found that total beta-glucan content in grey oyster mushrooms was the highest among of the three species. The amount of the another species within the white oyster mushroom and split gill mushroom, which is the first choice of edible mushroom species among the Malaysian consumer. So the cultivation of mushroom on oil palm frond can be the potentials to enhance the beta-glucan content in the mushroom fruit bodies, where it can play the role, especially for the functional food industry. So here is the acknowledgement. Here are the references for the studies. These are the publication and achievement from this project, and thank you.
Okay, thank you very much. Thank you, Siti Mariam, for the next presentation. So we have a question.
Thank you for the presentation, Siti Mariam. It's a very good presentation. So from your study, you say that oil palm frond have a lot of beta-glucan.
Yes.
And also have a high glucose fructose for the citation and reference, right?
Yes.
So can you go to the result again? So result of the beta-glucan from the mushroom. This is a discussion.
Yes.
So is this one the comparison that you plant the mushroom using the oil palm frond? Is it how you using the oil palm frond from your as a substrate? Is it you blend it as a material for mushroom media?
Yes, because this study I focus on the formulation productions, which I use my own formulation, and then I formulate the substrate using the oil palm frond to become the substrate for the growth of a tree species mushroom.
So you have your own formulation
Yes.
to develop, is it? So you don't buy or whatever, but you produce by yourself?
Yes, I produce my own self.
That's good, that's good. So it shows there's a good substrate for as a replacement.
Yes.
It's a good positive finding with the total beta-glucan. So you suggest that is it the fresh one or this one is a waste frond that you collect from the field?
This one is the waste one. waste that have available in the field, oil palm field.
Or you just collect all the front, doesn't matter the age of the oil palms.
Yes, doesn't matter. But for this one, I focused on the range of the front, means like from the like pruning process, it might be like
Pruning and harvesting all together, you
yes,
collect.
maybe.
So my suggestion maybe you can further study more, so that you can collect all different oil palm based on different ages, and then you can analyze those of fronts. I mean the compound, the psychochemical properties inside it, and then you can produce your substrate based on the ages, and you can test again which one is the best ages front to be used as a substrate.
Okay.
Perhaps you can find out because the oil palm farm at the 17 leaf at certain ages they have the highest amount of the physical chemical properties. Maybe this is my recommendation. But again, your result is already quite good. Perhaps it can be improved further. Okay, right.
Faiz. So I have one question. So is there any other methods to calculate the amount of beta glucan instead of you calculate calculate the alpha glucan first, then you deduct from the total glucan? Is there any other method that directly can calculate the beta glucan?
Because beta glucan is one of the polysaccharide, one of the carbohydrates. So in the carbohydrate, there might be like many types of carbohydrates. So we want to the specific parts of beta glucan. So we need to deduct other alpha glucan and all total glucan because they have like they maybe like sits in one group of glucans. So they might have various kind of glucans in that. So we want to have a specific beta glucan. So we need to deduct all alpha glucan and the total glucan, so that we have the pure beta glucan.
Okay, from your result, okay, so there is any significant difference between sodas, pedestral, oil palm fronds, and also within the 3 species of the mushroom? Because when I saw the value, it looks like close.
Yes. From the significant difference, because my one is I do like 3 flushes, 3 different harvesting periods. So from that, I do the testing among the species, and then they might be have like nearly same amount, but this oil palm frond is like bit higher compared to sodas and pedestral one.
You already analyzed using SPSS, right?
Yes.
So suppose you can see lah whether is it significant or not.
Yes.
Okay. Okay, thank you.
Okay, thank you, Ms. Siti Mariam Salama, for the next presentation. So with that, with the last presentation, we have the thank you everyone for the next presentation, and I think all of your research have a potential, and good luck for your undertakings. And thank you also for all the judges and also all the audience that stay until the final. Okay, so we have you all will stay because we will have other session, which is from a invited speaker, Prof. Dr. Syamsul Bahari bin Abdul Razak.
Hello, test. Okay. Welcome back all. Now I will invite our first virtual speaker, Professor Dr. Samsul Bahri bin Abdul Razak, a professor from the Faculty of Fisheries and Food Science, who joined us directly from University Malaysia Terengganu in the Zoom platform. A little bit about Professor Dr. Samsul Bahri. He is a professor from Faculty of Fisheries and Food Science from University Malaysia Terengganu. He graduated with PhD from Durham University. His field of expertise are biological science, botany, stingless bee behavior, meloponic culture, hevia biology, and microscopic techniques. He has been actively involved in several community-based projects to boost their socio-economic status, such as the integration of stingless bee in the stevia farming to produce a premium stingless bee, honey involving single mothers in Besut Terengganu, as well as the integration of stingless bee as a vehicle to assist smallholding in Malaysia. Now I would like to call upon Professor Dr. Samsul Bahri to deliver his speech. Please welcome.
Bismillahirrahmanirrahim. Thank you very much, Madam Chair Hussein, for a very nice introduction. First of all, I would like to thank you, the organizer, the Agro-Based Industry University Malaysia Kelantan, for inviting me to share some of my experience in developing community students in meliponic culture. So, Assalamualaikum warahmatullahi taala wabarakatuh, and good morning. So, I will share my screen. Share screen. Can you see the slide now? Organizer, can you see the slide? Is it okay? Hello?
Yes, hello. We can see it.
Okay. So my presentation today is stingless bee rearing as a potential activity to generate income. So my discussion topics will cover in this 20 minutes allocated time the Malaysian meliponiculture as emerging industry. Then I will discuss a little bit on the current status of meliponiculture. Meliponiculture is stingless beekeeping in Malaysia. And last but not least is whether stingless beekeeping a viable venture to be involved with. Stingless beekeeping in Malaysia is supported by mostly public interest. And as we can see today, a lot of public show their interest into stingless beekeeping. And the numbers of stingless beekeepers keep growing from years to years. This is partly because of dissemination of information through many platforms, through academia and community engagement, through media, social platform like, for example, Facebook. We have a lot of Facebook group discussing on stingless bee, WhatsApp group, hundreds of local WhatsApp group discussing on stingless bee publication through books and seminars, workshop, etc. And there are also almost all states in Malaysia, we have stingless bee association. For example, in Kelantan, we have drone nest, stingless bee association of Kelantan, Terengganu, Pelekat, in Pahang, and other state as well. So those are a platform where publics or members or people who are interested to get involved in stingless bee can be involved or get information from. And meliponic culture in Malaysia is actually an emerging industry because for the last 10 years ago, decades ago, we seldom heard about stingless bee. Not many people talk, not even from the public, also from the academia. And for the past 10 years, we can see a lot of interest and growth and abundant information keep piling into the internet. So it's very easy nowadays as compared to 10 years ago to get information on stingless bee. Even though it's an infant stage industry, you can call it industry, but it's grow very fast. We can see stingless beekeeping in terms of small activities, small ventures, medium scale activities, and also there are several commercial scale stingless beekeeping in Malaysia. And it is a very profitable activity, and this stingless beekeeping could be used to supplement income for stingless beekeeper. We can, if you go around Malaysia, if you go in Kelantan, for example, we can see a lot of stingless beekeeper rearing stingless bee and generate income from these activities. And they are also, these activities contribute to agriculture sectors, especially stingless bee is one of the beneficial insects, very efficient pollinators, and it pollinates a lot of our crops, not only in Malaysia but globally. And at the same time, become as stingless bee as a very efficient pollinators, they are also an agent to support the sustainability of our environment. And some people might ask, what are the differences between honeybee, lebah, and stingless bee or klulut in Malaysia? So as you see in the screen, on the left side is a picture of a lebah, a honeybee. The size is bigger, and on the right side is a stingless bee, the very common stingless bee, and the size is much smaller as compared to the honeybee. And honeybee, of course, they have sting, and they produce honey as well. Stingless bee, they don't have sting. Stingless, they don't have a sting, and they produce very good and nutritional honey. And little bit on stingless bee queen. This is just the introduction of stingless bee. And a queen, in each colony, we have only one queen, and the queen control. This is the only individual in this colony that control and govern the whole colony. So it's not the workers, it's not the drone or the male stingless bee that control the colony, but queen. And the queen lay eggs and produce workers, virgin queens, and drone. So a fertilized queen, like you see in the photo, they have a very engorged and large abdomen. They cannot fly after they've been fertilized by a male stingless bee. And this photo of worker, which is female, the one that you see flying outside the colony, they are all female stingless bee. So this photo show an example of female stingless bee, and this is an example of male stingless bee. So you can see a little bit in terms of their posture, right? So the male stingless bee, they looks more we can say macho in terms of their posture, right? So in each colony, there are approximately 10 to 15% of male stingless bee as compared to 80% female, because females, they are the one who do everything in the colony. And we are very lucky in Malaysia because stingless bee can only be found in tropical and subtropical areas, especially in this Khatulistiwa. But they can also be found in America, Africa, and Australia. So the map, the world map you see now, the green area, those are the places where you can find the distribution of stingless bee. And in Malaysia, we call it Indo-Malaya stingless bee. We have roughly now 80 species of stingless bee. Quite a number as compared to only seven species of honeybee. Stingless bee, we have approximately 80 species, but not all of these species can be domesticated and be commercialized to get to generate income. So these are the example of some beautiful stingless bee you can find in Malaysia. I just show you very quickly. Motrigona alici, fimbriata. These are very aggressive one, Lophotrigona carnifron, Collina, subterranean. And these two species, Heterotrigona itama and Geniotrigona thoracea, are the most common stingless bee species reared by our local stingless beekeeper. If you go around Malaysia, these are the two species that you are most likely to see in stingless beekeepers compound. And this just to show how beautiful the architecture of stingless bee. For example, this is Tetragonula melina. This is the entrance tube of Tetragonula melina. Very nice and very beautiful. And these are from Tetragonula ripeni, very colorful. And the color change every now and then, depending on the source of the pollen or the material that they use to build this entrance for the nest. So they will change color. This is Tetragonula testacitasis, very colorful as well. In UMT, we collaborate with Jabatan Pertanian Negeri Terengganu and we establish a repository of stingless bee in Sekayu. We collected 25 species of stingless bee and displayed it and collected it in this repository. But unfortunately, early last year, we had a very big flood. So now this repository is gone, no more. So we are going to we are establishing a new one in UMT second campus in Bukit Ko and ready to receive visitors. So you are most welcome to visit and see almost 20 species of beautiful stingless bee in repository Indo Malaya UMT at Bukit Ko. And all these stingless bee are documented in this book, Repository Clue Indo Malaya Sekayu, and also the story of expeditions, how we went around Peninsular Malaysia, Borneo to collect all those stingless bee, partly deposited in those repository. And we have organized almost 18 expeditions since 2016 until now, to not only to find new or maybe undocumented species of stingless bee, but also to meet the stingless beekeeper and to share information with the industry players. These are some example of stingless bee that are not found in Peninsular Malaysia. They can only be found in Borneo. Tetragonula melanocephala, Tetragonula sirindone, Wallace trigona incisa, Geniotrigona lactifasciata, Homotrigona hematoptera, Tetragonula rufi, Basalis, and this is a very unique stingless bee because we can only find this stingless bee with symbiont. It's a symbiosis with this Chromatograster. We can only find this stingless bee building their nest inside this end nest. So that's very unique, and particularly with this particular species. Another two species from Borneo. And okay, so those are a little bit on stingless bee, the introduction of stingless bee. And in University of Malaysia, we are actively involved with the community. My first project with single mother community in Jabi. So this particular project, we read stingless bee in stevia farming. So we produce a very unique stingless bee honey that contains stevioside. And we have published and analyzed the stingless bee honey and found the stevioside naturally, not by adding stevioside into the honey, but stingless bee itself produce this honey by foraging stevia plant. And also with the B40 group with ASNAF. And also in University of Malaysia Terengganu, we have a very specialized stingless beekeeping for student of UMT. We call it Kulupuno UMT. And we conducted for 1 year this program for 1 year, and we managed to have few alumni that still actively involved in stingless beekeeping. So commercial honey in the market, what are the issues? This is one of the big issues in not only stingless bee industry but also our honey industry, which is adulterated honey. So we have to be very careful when we go outside there and buy honey, because most of the honey sold in the market. In the market nowadays, we have record 80% of it are not pure, adulterated. So some people ask how these are very popular question. How we can differentiate between a pure honey and adulterated honey? So the only ways that we can surely or confidently have or answer that question is through lab analysis. And in UMT Malaysia Terengganu, we have established a very specialized lab, Akmal Quality Madu Pantai Timur, to help the stingless beekeepers analyze and then authenticate their honey. And we follow the Malaysian standard MS 2683 for stingless bee honey specification, and also MS 2679 for amalan baik on stingless beekeeping. And now we come to why some people want to get involved in stingless beekeeping. Firstly, very obviously, because if you compare to honeybee, they have no sting, so it's very easy to manage, and they are not becoming a threat to public. So stingless bee, they don't have sting, so small children, women, elderly can involve in stingless beekeeping. Easy to manage and very fast in terms of return of investment. If you start today involving or getting a colony of stingless bee, depending on your surrounding, it will give you can sell the honey next month. So it's very fast as compared to other agriculture-based activities, and you can get repeat income. I always told our stingless bee friend, getting involved or having a stingless bee in our compound is just like having an ATM machine. Whenever you want to get some money, you harvest the honey and then straight away sell the honey and get the money from that selling. So it's like ATM machine beside our house, and it's very high volume product, because we know honey also very nutritional, being published in many scientific journal, and they give a good commercial value in terms of products. You can produce not only 1 or 2 products from honey, from propolis, or from bread, but you can produce thousands of downstream product as well. And stingless bee, because they are unique, we can only find it in tropic region in our country. It's very unique. European, they don't have stingless bee in their compound. They cannot, if they want, even if they want to raise stingless bee, they cannot do it because of the distribution. Stingless bee cannot thrive in their temperate region. So we are very lucky and unique. So those are also one of the selling point of stingless bee if you are thinking of getting into stingless beekeeping. And the price of stingless bee colony, meaning that if you want to start your stingless beekeeping, this is the price or the costing that you need to take into consideration. Nowadays, you can get 1 colony, the sarang of stingless bee honey, stingless bee sarang, it's around 500 to 650. That's the average of 1 colony cost nowadays, which is depend on the location, the strength of the colony, and the type of the species of the colony. So I think this is if you want to have like 10 colonies, you have to have like 5,000 ringgit to get 10 colonies of stingless bee. And stingless bee, they don't produce honey all year round, so we have to take that into consideration as well. So starting from October to January or February, stingless beekeeper, they don't harvest their honey. So the honey flow seasons start from March to October. So those are the time when stingless beekeeper harvest and sell their honey. But during those, we call it death season, unproductive season for stingless beekeeping. stainless beekeepers still can get their income through the production of downstream product. For example, cosmetic, like we see a lot of people selling a soap, soap, honey soap, propolis soap, lotion, cream, a thousand of others cosmetic product from stainless bee product. Okay. And these are example of varieties of other products, madu keluak bawang putih, garlic, acar ikan masin, madu keluak, cookies. So many other product you can think. There are limitless of product you can imagine. It's up to you. These are also another example of stainless bee honey. Different packaging, different labeling, and the income of roughly this is an example of income generation from 50 colonies from the stainless bee single mother community in Jabi. So roughly 1 year they managed to get 1 month they managed to get 1 year average 20,750 ringgit per year from that 50 colonies that they read in stevia plantation. So normally they will sell kelulut honey. There are many platforms to sell kelulut honey. So you can sell it direct from beekeepers. You can collaborate with the association or club, can do contract farming, and also very popular. Most of stainless beekeeper now, they market their honey through e-commerce like Shopee or Lazada. So I publish several books on stainless bee honey, stainless bee. You can get the books from University Malaysia Terengganu, or you can contact me if you have you want to get more information, or if you want to visit UMT, or you want to visit do collaboration with UMT or my colleagues in UMT. We have 18 researchers in UMT that involved in stainless bee research. So we have special interest group in University Malaysia Terengganu that focus on various aspects of stainless bee. So with this presentation, I would like to thank you very much for your kind and your passion listening to my presentation. Thank you very much.
With Professor. If anyone have questions, you can ask.
Hello. All right. Hi, Prof. I'm Raimi from Faculty Industry Study. My question to you, what about the storability of the honey from the kelulu? Is it the same as the normal honey? Because I actually heard some people actually said that if the honey is actually stored for a longer term, there is some compound in the honey that can be harmful, as it can be carcinogenic. Can you give some insight on this? Thank you, Prof.
Okay, thank you very much for the question. As you know, stingless bee honey, they contains a lot of beneficial microbe, especially yeast. And these microbe, they are very good, especially for our digestive system. And you know, yeast, they do the fermentation, and that's the beauty of stingless bee honey actually, because as compared, we have several publication on the stingless bee honey composition, and we showed that the composition of beneficial microbe in stingless bee honey is way much higher than as compared to in honeybee honey. So in terms of shelf life or storage of stingless bee honey, it is now still recommended to store as soon as possible after harvesting your stingless bee honey in the chiller or refrigerator, for the reason that we want to reduce the activity of this beneficial microbe. So it's a bit different as we compare to honeybee honey. And in terms of carcinogenic compound in stingless bee honey, I think you are referring to HMF, right? The composition of HMF. So HMF in the pure or natural honeybee, as for example, as soon as we harvest stingless bee, the composition of HMF is almost zero, and it will keep increasing. The HMF will keep increasing by exposing the honey to the UV, to the light and heat. We know Malaysia is very hot, so that's why we always recommend stingless beekeeper to immediately after harvesting keep their honey in a refrigerator. First, because we want to reduce the activities of this beneficial microbe, and number two is to slow down the formation of HMF, and because HMS also will be produced after time. I mean, if we keep the honey for a very long time, and what I mean very long time, take years, right? Not take days, but years. The HMF will become the composition of HMS also will keep increasing, but not but still the limit, the composition of HMI still under the limit of allowable by MS standard. Does it answer your question?
Other questions?
Okay, I have one question, Prof. My question is that what is your opinion on the effects of the climate changes? How this climate change affect bee farming? I mean, stingless bee farming. And my second question is that what is the dose, effective dose when it is incorporated in healthcare products or medicinal products? Thank you.
Thank you very much. Yes, obviously, global warming or climate change has a very adverse effect on not only on bees actually, but also on everything on Earth. So, for example, we talk about bees in particular, right? Bees, they associate, or we closely associate it with the surrounding, and they get their source or their livelihood through foraging on plants for their nectars and pollen. And we see the pattern nowadays, even on a very short period of time, we see that the pattern of flowering plants, right? We see now, for example, durian, rambutan, they are not flowering according to the previous seasonal flowering time, right? So this make the bees confusing. So those are the things that affect stingless bee, and also throughout global warming, we see a lot of natural disaster, and especially through deforestation. So those are the source, the main source of not only for bees but other beneficial insect livelihood. So in particular, in an essence, global warming, climate changes has a very adverse effect on stingless bee in particular. And on your second question on the dose, right? On the dose. So because bee or honey, honey is not considered or not accepted as of medicinal or medicine in modern medicine. It is in alternative medicinal categories. So we don't have actually the exact dose to be consumed by users, because even us as a human being, we have a very physiological differences from each individual, right? So but the most common practices in taking stingless bee honey or honey is one small teaspoon in early in the morning and before we go to bed. So those are the most common practice of consuming stingless bee honey as a supplementary food. And if you want to make it into product, I myself are not is not an expert on those product. We have team members here in the food department that produce many innovative product using several products from stingless beekeeping, honey, propolis, and bee bread. Maybe if you are interested, you can contact my colleague in the food technology department. Okay.
Any last question? No more questions. Okay. Thank you for the all the questions, and thank you, Professor Dr. Samsul Bahri bin Abdul Razak, for the talk. Now is the second session for the oral presentation. I will pass to the moderator for this session.
Okay, Assalamualaikum and a very good afternoon, everyone. My name is Nordini Rusli from Faculty of Agro-Based Industry, and I am your moderator for this session. I would like to welcome all participants for the physical presentation session 2 at Seminar Room, Library UMK. And I would like also to welcome the judges, Associate Professor Dr. Lee Seong Wei and Dr. Mijanur Rahman. All right, I think let's get down to business. Before that, I would like to kindly remind all participants that time allocation for oral presentation is only 10 minutes. So maybe you need to spare at least 2 to 3 minutes or 5 minutes for Q&A session. But if we exceed the presentation time, if there is any more question, you may give the question to me so I can ask the presenter directly. Okay? All right, so without further ado, let me call upon the first presenter, Binti Mohd. So she will be the histology of Oreochromis species. Please welcome Ms. Munira Binti Mohd from the School of Agri-Technology. Okay, I think we just proceed to the third presenter because Munira and Rivitra are actually absent for certain reason. So I would like to call upon Ahmad Rahimi bin Razli to present his presentation on the effect of pelleting the creep and growing ration on its chemical composition. Please welcome Rahimi.
Yes, yes. Okay, hello everyone. Very good afternoon to everyone. So I am Ahmad Rahimi. I will present about my study, which is effect of pelleting the creep and growing ration on its chemical composition. So the introduction to total mixed ration, which is TMR. So total mixed ration is a chopped green fodder or silage blend with cereal, cereal byproduct, protein source, minerals, vitamin and feed additive. So here I list two types of TMR, which creep feed for pre-weaning, usually for sign-in goat, growing ration and post-weaning, also for sign-in goat. This TMR act as a supplement for the kids and lambs. It's really good. TMR can boost their growth, immune system, and good quality of carcass. So for the objective, first to assess the chemical analysis of local feed resources for formulating creep pellet and growth pellet on the pre-weaning and post-weaning of sign-in goat growth performance, to evaluate the effect of pelleting the TMR for pre-weaning and post-weaning sign-in goats on its chemical composition, and lastly, to analyze the significant difference between the creep pellet and growing ration in the form of mesh and pellet. So for the methodology is we do the proximate analysis, which is determine the crude protein, crude fiber, ether extract, ash, and dry matter. Okay, so for the analysis of nutritional value of TMR, which is it is the result for the proximate analysis for the pre-weaning. So here we got mesh form, got the 19% of crude protein, and pellet form is 22%. So for the ether extract, which is crude fat, we got 1.3% for mesh and 3.53% for pellet, and so on. Okay, this is the result for the proximate analysis for post-weaning. For the crude protein, we get the lower from the pre-weaning, which is 15.51% for mesh form, and the pellet got 14.31% for the crude protein. Okay. So the statistical analysis for my study, I use the student t-test, which I compare between mesh and pellet for both pre and post weaning. So this is the result for the crude protein, which you can see it is no significant difference between mesh and pellet. It is same for crude fat also, no significant difference. Dry matter also 0.07. Ash 0.246. So for the crude fiber, we had a little bit problem which the machine cannot run properly. So we get only 2 duplicate sample only. So when we do the test, the t-test, it cannot provide the significance between them. So this is result for post-winning protein. So it is significant difference 0.04, and crude fat got no significant difference. Dry matter and ash. Okay, so for the conclusion, based on the data, it was shown that the chemical composition of creep feed and growing erosion has no significant difference between mesh and pellet, except for crude protein for post-winning. So this TMR give a rapid growth for the goat during early life, can minimize the cost of rearing, and thus can provide a more profit to the farmer. Next, improve feeding efficiency, better than conventional feeding, which animals likes to sort during their feeding. So, and lastly, use of the local feed resources that always available around us can substantially reduce the cost of production of livestock. So that's all for me. Thank
you. Okay, thank you, Rahimi, for your presentation. From your presentation, I can see you are going to making the pellet,
yes.
pellet, and want to compare with mesh.
Yes.
So can I know why for goat feeding we need to make pellet? Because the conventional method is the TMR, like a mixer, total mixed ration. So why we need to make pellet for goat feeding?
Okay, why we need to do to make pellet for the goat? Because pellet can increase the feed intake for the goat and provide the enough nutrition also for the goat.
And another question you mentioned in objective 1, the growth performance, and also you are making conclusion, increase the growth performance. But your materials and method or result discussion doesn't indicate about the growth. So why you make the conclusion, TMR give a rapid growth? Because there is no data.
Based on the conclusion, that I provide the TMR give a rapid growth for the goat. So
I think since you don't have data, so cannot make the conclusion about that. So your data is only chemical composition. So you are comparing with the mesh and pellet, and you found not significant.
Okay.
So I think this is like proposal. Means you're planning. Later you want to do on growth performance. Is it?
Yes.
Yes. So in that case, you need to show the like Gantt chart, the milestone.
Okay.
So maybe, yeah, in your presentation. Yeah, like Gantt chart, expected outcome and milestone. Okay, then. So I saw from your result the protein content or ether extract content, even though not significant, but it was a trend to increase in pellet. So what is the reason? Compared to mesh form, you can see the pellet, they contain more protein, more fat. Why? Because ingredients are same.
Sorry.
So you can see, can you show me the table, the chemical composition of the mesh and pellet?
Oh, this.
Yeah, the previous one. Yeah, like this one. You can see compared to mesh, pellet contain high protein, high ether extract, high crude fiber, high dry matter. Why? So do you have any comments on this?
It shows that every proximate analysis got the higher percent of the nutrient. As we use the right ingredient that can give more percent of the crude protein to the animal.
Okay, Mr. Rahimi, so you are working on feed, feed.
Yes, sir.
Got feed. You do compare between pre-weaning and also post-weaning. So one thing I concern is pre-weaning is they still rely on the milk. Am I right? So any previous study that can support your research? Any previous study they have working on this comparison between pre-weaning and also post-weaning? Because pre-weaning, to my understanding, is they still rely on milk. So they can choose not to eat, not to consume the formulated feed. Rather than what I concern is why you compare to the pre-weaning. Any previous study can support your research methodology. This is only one thing that I concern.
Okay.
Dia macam kita compare bayi yang still menyusu dengan kanak-kanak yang makan makanan kita lah. Faham apa saya nak sampaikan? Itu yang saya concern. Kalau ada previous study yang buat kaedah yang lebih kurang macam Cik Rahimi buat ni, saya rasa tak ada masalah. That's all saya concern. So mungkin ada orang kan question, kenapa kamu compare dengan ni pre-weaning, they still rely on milk. They can choose not to eat. Am I right? This is my big question mark. Okay lah, this is not question. You no need to answer. I just concern. Saya ambil berat je, tak perlu jawab pun.
Thank you, Doktor.
Tapi kalau ada kajian yang lepas menunjukkan dia buat pembandingan sedemikian, so tiada masalah lah.
Okay.
That's all from me. Thank you.
So is there any question?
All right, thank you, Mr. Rahimi, for the presentation. So I think that we just proceed to the next presentation. So I would like to call upon Miss Fatin Atira binti Muhammad Sabri and she will be presenting the sexual behavior of male goats in different isolation period. Please welcome.
Assalamualaikum warahmatullahi wabarakatuh. My name is Fatin Atira binti Muhammad Sabri, and today I will present about sexual behavior of male goats in different isolation and period. The research background. The behavior pattern of an animal is important to identifying their needs in order to increase their productivity. And the temperament of an animal, especially in goats, can determine their social isolation, and the activity and placement of the goat can affect sexual behavior, and sexual behavior can affect the semen collection because semen collection requires a male to mount a female in order to ejaculate inside the artificial vagina. And that is why the goat must be separated from the female to increase their sexual desire. And some researchers stated that social grouping and day length are the causes which can influence the animal behavior. Problem statement. Most farmers mix male and female goats together in the same place. And when the male goats are making natural mating anytime when they want, this can cause the difficulty to perform the semen collection with male goats that has less sexual desire. And the objective of this study is to determine the reproductive activity on different isolation period in male goats. For the method, a total of 4 boer male goats and 6 female goats was used in the study, and each group consists of 2 boer goats and 3 female goats. And there was 2 groups. There is Group A from local university and Group B from private-owned farm. The differences between these both groups is Group A, where the goat was separated individually in a pen for 28 days, and Group B was separated individually in a pen for 14 days. The female used in the study were synchronized for estrus period, and the semen collection was performed by using artificial vagina and during the day at 9 a.m. The experiment was repeated 3 times, and the reproductive activity data recorded was semen volume and sexual behavior such as anogenital sniffing, mouth with intromission, phlegm, mouth attempt, and self-urination. Mouth with intrusion is where the penis entering the vagina during a mount, and phlegm is where it is a behavior response that male detect particular smell from female, and the characteristics is by curling of upper lips and raise their head. Results and discussion. Table 1 shows the semen volume and percentage of the success collection in both groups. And as the result shows that there was no significant difference on semen volume between both groups. As you can see in the table, semen volume for group A is higher than group B, but there was a significant effects on success rate of semen collection between groups. This is made due to the behavior pattern that causes by day length and social grouping. Johnson stated that size of social group and sex of the goat affect the behavior, while day length was important in entering the mating season in goat. And as you can see in the results, the success rate of group A is more than 19% than group B. This can conclude that group A has high sexual desire than group B. It comes to agree with Chancer that stated that time of male goats isolated from their goat herd for 4 to 6 weeks affected the reproduction mechanism of goats upon reintroducing into the herd. Next, figure 1 shows the difference of sexual behavior between group A and group B. The results show that there was a significant difference between both groups in terms of inogenital sniffing, mouth intromission, self-urination, and phlegm. Fernandez stated that social separation in sexual inexperienced photostimulated male housed individually influenced their locomotion, extenuating inogenital, which were activity involved high motor skills. And there was no significant difference in mountain, which agree with Fernandez that state photostimulated male house individually and in group have similar mountain. Sexual behavior in individual house inexperienced photostimulated male were less intense rather than in a group, but it does not reduce their ability to induce sexual desire. For the conclusion, we can conclude from the study that the day length and social grouping are important for their sexual behavior, and the male goat with isolation period of 28 days have higher sexual desire for mating rather than male goats separated for 14 days. And the overall conclusion that there was a significant difference in sexual behavior between groups with different isolation period. That's all for me. Thank you.
Thank you, Fatin Atira. Have you been missing your supervisor?
Huh?
Are you miss your supervisor? Do you miss your supervisor?
Yes.
All right. Is there any question from the judges?
Hello, Fatin. Thank you very much for your presentation. So you mentioned that Group A and Group B, they got from different farm. Like Group A, local university. Group B from private farm. May I know why you choose different group, different place?
Because the limitation of the animals itself.
Okay.
At the local farm, there's only two males, and we want to differentiate between male and not going to use the same male. That's why we're using the private owned farm and a local university.
Yeah.
Yes.
So you mentioned local university for 28 days.
Yes.
And private farm is 14 days. Do you think that one also one of the factor that can affect on the result?
Yes.
Because different place, different house, different environment, so that can affect on the sexual behavior.
Yes, because the social grouping and day length and some of it is the placement itself is affecting the sexual behavior, and sometimes private owned farm, they just mix together between male and female goats. That's why we want to see if there's any difference if you separate it for 28 days and for 14 days. And usually private owned farm, they didn't separate the goats that can affect if we want to collect the semen collection. We want from the semen collection.
I got it, but I am asking, you got the difference, significant difference.
Yes.
That one not because of 28 days or 14 days, maybe because of the environment, because different place. Is it? Or
Yes. That may be because of environment. Just yes, because the different how they manage their goals and the temperature during that time. Yes, it's maybe to the upper cause. The main of this study is want to see the behavior and the separation. So about the effect on the environment, I'm not sure about that, but I can further investigate about that.
Okay, Miss Fatin, Atira. So your research is about to study the behavior, sexual behavior of the male goat. What is the species?
Bowl.
Oh, bowl. So you need to mention what is species in the title basically. So my concern is your study is show 28 day is the best to make the sexual success. But I think you should look any control in your research. Control. Like I'm not animal science based research, but what I concern is up to date or at present, people will look at the artificial insemination method instead of waiting for 28 days to reproduce for the reproductions activity of the goat. So maybe you can include the artificial insemination method or technology in your research to make your research more orang kata up to date lah, up to date. Because as a farmer, I think it's not logic lah. They need to wait until 28 days because it's too costly. It's not cost effective. If I'm a farmer, I will rather choose artificial inseminating rather to waiting for 28 days or 40 days. So this is my comment. You no need to answer. This is one concern. So make sure your research is up to date. Up to date. Thank you.
any question?
I have a question. I just wonder, how did you analyze your data? What kind of test that you use? Because like what Dr. Raimi concerned is about the factor, another factor, which also include the environment.
I'm using t-test.
So you Mm. And then different isolation period as well. So that you have so many factors, which I don't think you can use student t-test. Yeah. So, yeah, another two independent variables. You know what I mean? So maybe you need to look back at your statistical analysis. Otherwise, your data must be like haywire sikit kot. Okay. So, I'm not
Yeah,
saying it's wrong, but maybe you need to double check.
Okay.
Yeah.
Okay. Okay.
Okay.
Okay, thank you.
So that's all from me. And is there any more question from the audience? So the students also are free to ask question as well. No, okay. So thank you so much, Fatin Atira, for the nice presentation. So let's move on to the next presenter, which is Nur Farahi Nabila Binti Muhammad Shah. So she will be presenting the effect of pelleting on the shelf life of total mixturation for lactating dairy goods. Please welcome.
Bismillahirrahmanirrahim. Assalamualaikum and very afternoon to the all panel and the audience. Okay, my name is Farahi Nabila Binti Muhammad Shah. Today I would like to present part of my study, the effect of pelleting on the shelf life of TMR for lactating dairy goods. So this is the content. For the introduction, Asia has the 146 indigenous goat types that accounting 26% of from the worldwide, and only 9% of this breed exists in Asia. And in 2017, Asia had the largest proportion of the world population compared to other regions in the world, like have the figure in this slide. So for the industry, in Malaysia, in 2005, Malaysia has a small goat population compared to another region in Asia, while in 2007, the Malaysian government have supported the import of live goat from the other countries. And in 2021, the data showed the number of goat have increased from 2022, and Kedah have been contribute to this value. And from the 5 years previous, the imported live goats will slowly drop from 2016 until 2020. For the goat meat production, it is inadequate, informal, and very fixed for the data. So this is the subject that I will use in this project, which is the Sanin goat. That really we can see that they have the white for the body color. And why I choose this subject is because of they have the heavy milk producer. So we back to the topic of the pelleting and shelf life. They have the relationship where by the pelleting, the heat that produced from the pelleting process will increase the storage value, as the heating process will kill some of the bacteria and remove the moisture. Also, by the pellet that produced is completely dry, it will less prone to the spoilage and shrink. So the relationship by the lowering the moisture, this will increase the shelf life of the pellet. So for the problem statement, in the market, there are variation of quantity and quality of pasture feed, but it is very difficult to meet the demand at dairy production stage, which at the production stage of milk, the goat need the balanced nutrition to produce the milk. By the nutrition imbalance, this gives negative impact on health, fertility, milk production, and growth performance. So the current study by Mira et al. 2022, they have successfully produced the TMR, but it is in loose form. So the problem by the loose form, the animal behavior can sort what they want to eat and consume. So by this, they can affect to the goat's body requirement. Also in the loose form, they contain the high moisture content. So the pellet can be easily spoiled because the fungus can be grow easily. Also, it can disturb the storage by the overheat from the moisture. So from that study, this project have continue because the TMR feeding TMR feeding provide the better system compared to conventional feeding, especially in terms of dry matter. Also, the TMR pellet have commercial potential in future because of contain low moisture. Also, in the available market, mostly the pellet are in grain-based ingredient. Also, by the TMR pellet, it can provide the better flow for feeding where the lower cost and labor, also the wastage and dust. So the hypothesis of this study is the chemical properties of for the hash null, the chemical properties of the MR pellet have a significant difference between the wicks. By the hash alternate, the chemical properties of TMR pellet have no significant difference between the wicks. So for the objective, first is to assess and compare the chemical analysis of local feed resource for formulate TMR, pellet TMR, and loose TMR. Second, to evaluate the mineral properties of pellet TMR for lactating goats. For the methodology, from the objective, we come up with the number first, the chemical analysis, which we have used the proximate analysis by standard method by AOAC for the crude protein, crude fiber, either extract, ash, and dry matter. While we go to the second objective, we have for the mineral analysis by using the acid digestion and ICP-OES method. We analyze the calcium, phosphorus, potassium, and sodium. So for this project, it can be divided to a few parts. First is development of TMR formulation. Second, conversion of loose TMR. Third, to the improvement of TMR quality. Second, the feeding trial. Fifth is the data collection and statistical analysis. For the outcome, this is the result proximate analysis for the raw ingredient that I have used for formulate this TMR. Then for the shelf life, this is the result for shelf life for TMR loose and pellet, where the loose TMRs show they have the significant difference have in the either extract and dry matter, where for the pellet only crude fiber show the significant difference. This is the result for mineral analysis for TMR loose and pellet, which show the pellet TMR show the highest result compared to loose TMR. The reference, and thank you for the attention.
Thank you, Faraheen. So I welcome the judges and the audience for questions.
Okay, thank you, Faraheen, for your nice presentation. So, your research is about making a pellet for goat feeding. So, goat, this one is post-weaning.
For lactating.
For lactating, okay. So, is there any research on pellet feeding?
Yes.
Have been done earlier by other researchers?
They have done for the pellet MR, but for the fattening lamb.
Yeah, but in your research background, you did not mention about that one, about pellet feeding on the goat.
On the goat, they have less study for pellet TMR. So this is like I mentioned earlier. This project have come from the TMR from the MIRA 2022. They have successfully produced formulation by using the fresh napier to come up with the loose TMR.
Yes, based on literature, farmers are using loose TMR or silage TMR, but not many literature about the pellet feeding.
Yes.
So you also mentioned about the disadvantages of loose TMR or TMR silage. But how about that one? If you give the pellet to the goat, so ruminant usually they eat 24 hours all the time. But if you give pellet, within half an hour, they will finish the pellet. So other time, what they will do? What is your opinion?
In this project, I focus to my project. I have provided the supplement to give activity after they eat the pellet. Also, we know that the ruminant need the time to the digestion for their ruminant digestion. So they will take time for their time.
Okay. So I think what is called animal behavior study also need to conduct in this study, because ruminant they need to be involved 24 hours with the activities. So if they take pellet, within 30 minutes, 1 hour, they will finish it.
Okay.
So we need to give something like UMMB or other some supplement so that they can take it. And also you mentioned you want to observe meal production or so many parameters, but this is like ongoing.
This is ongoing because now I'm done for the adaptation period only. So we need a feeding trial to know the performance of their growth.
So better to show your like milestone or research activities in future.
Okay, doctor.
But you did not mention about that. Okay, can you show me the fruit fiber content in pellet? So crude fiber is the 24. So ether extract fat is increasing.
Yes.
So what is your opinion?
I think because of the pelleting process that we use PFAD in this formulation, they have absorbed to the material, the ingredient have been mixing well because due to the longer time take in the shelf life.
And another question, this one is preservation method, keeping quality, means, so you observe only until week 3.
Yes.
So is it okay until week 3, I mean 21 days farmers want to keep in their farm? Not longer than 3 weeks?
I will go with another week after this. So until now, the data that we have only until week 3.
Because for commercialization of the pellet, farmers need to keep at least 2 months.
Yes.
2 months, 3 months. So if you observe only 3 weeks, not practical.
Yes, I know.
Okay, thank you.
Okay, thank you, Ani. So now Farahi Nabila, Muhammad Shah.
Yes, doctor.
So you're working on to check the nutrition loss due to the time. So how long you planning to do overall for your research?
After this, I will go continue with the feeding trial. That take 90 days.
90 days.
We will check the growth performance
3
of the
months.
goat from the body, blood, and their feet. We have done earlier.
Okay, I understand. So I think just now I so concerned about the comparison duration between week. Yeah, you actually should carry out between months because it's more significant if between months, and you need to do market survey. Yeah? Market survey to check what is the shelf life for the commercial pellet in the market. What is the shelf life?
Shelf life is usually take the 3 months to 6 months.
So you need to check lah. Your formulation is can sustain or can stand up to 6 months lah. So my second question is I want to test your knowledge. So just now you have mentioned fungi contaminated feed is harmful to animals. So what kind of toxin they produced by the fungi? They are harmful to the animal.
Acidosis.
Acidosis? No. I think you need to do study a lot. Okay, that's all for me.
Okay, thank you, Doctor.
Is there any question? No? Okay, thank you.
Thank you, Farahin, for the nice presentation. Okay, I think we just come to towards the end of the session. So I would like to welcome Ms. Nur Naziha Amira binti Muhammad Suhaimi to present on the comparison treatment between SIDA and herbal in detection estrus signs and follicle size of the female goats. Please welcome.
Okay, Assalamualaikum and good afternoon. My name is Nur Nazifa Amira binti Muhammad Suhaimi. Okay, I'm supervised by Dr. Jeli Irina Raja Khalif. So today I will present to all of you about comparison treatments between CDAR and herbal in detection estrus signs and follicle size of the female Boer goats. Let's move to introduction. Parallel advancement in livestock was arguably insufficient. It may harbinger of inferior reproductive performance and scarcity of high producing breeds. So the self-sufficiency level in Southeast Asia is very low, optimizing low breeding in Malaysia. That's why, as you can see, only 11% the self-sufficiency level in 2019 compared to Indonesia and Philippines. A deliberate policy designs in cattle production has gained legislative consent and towards emerge as importers of goats breeding. That's why we can see that Malaysia is very low compared to other Southeast Asia. Okay, low in reproductive performance can make the Malaysia the first ranking that in Southeast Asia that import goods compared to other country in 2019, which is you can see that more than 200,000 tons that have been imported. So implementing progesterone as an animal estrus inducer for estrus synchronization can decipher the poor heat detection. Then control internal drug release have been used previously to make the animal to come into heat. However, it have like a few drawbacks. For example, it's a high cost and practical. It may require the labor skills and also hurting the animals. So we have been made an innovations we call as herbal Pregnomix. Pregnomix is an orally feed supplement that consists of ginger, cinnamon, and also black pepper. The components of Pregnomix is ginger, or we known as gingiber officinal. It may sexually regulate the sexual activity and also GnRH. The second one is black pepper. They may have raised follicle stimulating hormones and the number size of Leydig cells. And the third one is cinnamon. It may improve norepinephrine, and which is norepinephrine can increase the release of nitric oxides to stimulate the hypothalamic and active the gonadotropin hormone. The objective of the present study are to measure the expressions of estrus signs in female goats. Second, to determine the follicle size at the onset of treatments to ovulation. Let's move to the literature review. If you wonder how the herbal can interact with the reproductive systems, the first one is because herbal is considered as an alternative to hormonal treatment in order to increase the reproductive efficiency. The greater dietary intake may increase FSH concentration, and it may associate with increase in ovulation rate. If we are feeding the goats with the sufficient of animal feeds and supplemented with herbal, this dietary intake can increase the FSH and LH, and also fecundity rates. The ovulation rate also could be increased by high energy and high protein supplementation in animals. This is a list of active ingredients that cause or we call as a trigger the animal to come into heat, which is piperine. For the black pepper, ginger, we have like 6 gingerol, and cinnamon, we have cinnamaldehyde and cinnamic acids. Next is research methodology. I've been done these experiments at Kaprima Hulu Seladang Valley Farm, Terengganu. The numbers of animals involved is 24 female goats, boer female goats, involved with the 3 groups. First is herbal, or considered as a control, then herbal plus vitamin E and also cedar. The criteria of selection would be good reproductive health, adequate body condition score. And then this is the herbal preparation, which is all the herbs were ground using a grinding machines and were packed in a store airtight sample containers for proximate analysis, and then we will be done at the female goats. This is a dietary intake as because it's involved with the formulation, so we need to know their dietary intake for the energy and protein, because both of them were very important to know to maintain their requirement, like for the both of the growth, for their maintenance and average daily gain. So we calculate their dietary intake, and we know that how many they need for energy, for their proteins. And this is the protein requirements. This is a ratio formulation that we have done before we mix with total mixed ratio. We call it as a TMR. Then this is experimental designs, which is treatment A. The herbs, 8 female goats were fed with herbs mixed with TMR for 7 consecutive days. Treatment B, cedar, implemented for 18 days, and treatment C, herbs plus with vitamin A, and then fed herbs with for 7 days. And then we observe for the behavioral manifestations of estrus signs via visual observation for 45 minutes. And then behavioral manifestation, for example, we have like tail wagging, the redness of vulva, standing head. Then the follicle size were identified starting on day 0 until ovulation. The outcome and measures and analysis. The chapter 4 results. This is estrus expressions exhibited by goats. You can see that herbs plus with vitamin E is the highest estrus expressions, which is 72.92%, followed with cedar and herbs only 41.67%. This is estrus exhibited by goats. Then estrus signs response exhibited by female goats, which is herbs plus vitamin E, was the highest at 72.92% compared to herbs and cedar at 41.67 and 64.58% respectively. So one of the evidence I can say that cinnamaldehyde, for example, cinnamaldehyde is the active ingredient that contained in cinnamon. It may derive from cinnamon that improve norepinephrine. So it may increase the release of nitric oxide to stimulate the hypothalamic axis and active the gonadotropin hormone. This is a follicle diameter of the goat follicle from onset of treatments until ovulations. The mean diameter of ovulatory follicle observed in herbs plus vitamin E was the highest at 9.0 mm, followed with cedar at 8.76 mm, and herbs only 8.50 mm. So deep ovulatory follicle diameter synchronized was larger than Buchar et al. In boer goats at 7.61 mm. So as a conclusion, as you can see previously, herbs plus vitamin E was comparatively the highest, achieving the satisfactory results for the estrus signs and follicles compared to herbs only and cedar. So therefore, in meticulous attention to detail, we need to give a proper attention to herbs plus vitamin E because it may provide a novel opportunity for estrus synchronization program. Thank you.
Thank you, Nazifa. That's a nice presentation. I believe that you are towards the end of your study.
Yes.
All right, okay. I just have a quick question regarding the formulation for herbs plus vitamin E. So what kind of vitamin E source that you use?
Vitamin E selenium. I get from a veterinarian.
Okay. Does it cost a lot? How
No,
about
a bottle costs about 60 ringgit. It can be used to 10 female goats.
Okay. Did you really count the cost-benefit ratio between those different treatments?
Yes, because if you use cedar, it may cost like 400, right? But if we use herbs plus herbs plus vitamin E, it's only cost like 10 ringgit per use only. So it's a very beneficial and cost effective.
Okay. All right. Okay. How about the herbs itself? Because those herbs also, quite costly.
The herbs because it's only 41.67%. We need like to formulate again to make it better, to perform better for the next.
Okay. Maybe I will ask the judge, any other judges to.
Okay, thank you, Nazifa, for your nice presentation. So I want to ask, what is the dose of the herbs? Dose,
yes.
herbs
Okay.
per goat, daily.
Yes, 0.2 gram per dose per head per day.
0.2 gram.
Yes, per head per day.
So 0.2 gram you mix with other
TMR.
feed or TMR?
Yes, total mixed ration, TMR.
And you continued for 7 days?
Yes, for 7 days.
What is the successful rate for showing the estrus?
Showing the estrus, the successful rate, 72.
Is there any data?
Yes.
I want.
20. 72.92%.
Can you show the table?
The table.
Okay. So, but here don't have successful rate. Don't have?
I mean the average is the successful rate for the degree of estrus expression.
Average.
Yes.
Okay. How about the pregnancy rate? The pregnancy rate.
Pregnancy rate? No, we didn't measure until pregnancy. Until this.
So after show the estrus,
yes,
you did the natural mating.
Yes. Actually,
but after natural mating, not continue.
Yeah. Actually, I also withdraw their blood, but actually I didn't complete yet the analysis.
Okay. So if no pregnancy rate, how we can ensure animal become pregnant or not?
Oh, because we measure their ovulation rate. The ovulation, the follicle size.
So follicle you measure using
ultrasound B mode ultrasound scanner.
ultrasound
Yes.
scanner. So you only measure the follicle size.
Yes, the follicle size.
So follicle have primary follicle, Graafian follicle.
Yes, Graafian
So what
follicle.
follicle?
It starts from the secondary follicle because it starts from day 0, secondary follicle, until day 10 it becomes corpus luteum.
Okay, okay. Okay, thank you.
Okay, no Nazifa.
Yes, sir.
No Nazifa Amira. So your supervisor is leaving for postdoc.
Yes, in Belgium.
Actually, you need, based on the regulation, you need to change your supervisor.
Oh.
Based on the regulation, because she on leave.
Oh.
So any signature approval during her postdoc is not valid. So please take alert. Ni TDBI. Okay, ni no Nazifa.
Yes.
So you are doing on herbs.
Yes.
Plus fat soluble protein, vitamin E.
Yes, vitamin E.
So your research show that the herbs plus vitamin E perform a better result.
Yes.
So how about others fat soluble protein instead of vitamin E?
Because vitamin E it's contain anti-estrogenic activity. Yes.
How about vitamin A, B, K?
We didn't try it.
How about you don't do literature review?
Yeah, because I say that vitamin E is affect the follicle. It's affect the follicle size compared to vitamin A and so on. But vitamin E is like they have cited papers that mention that vitamin E perform on follicle. So that's why I'm using vitamin E.
Okay. So you know the mechanism and mode of action of the vitamin E. So how about the mode of action of the herbs?
The mode of actions of herbs. Actually, I've done HPLC, high performance chromatography, but I didn't put here. Okay, actually, when I'm doing HPLC, I take their blood. During the goat estrus, it contains piperine, and it means that piperine trigger the animals to come into estrus. Yes, herbs that works very well.
Okay lah. So please avoid the old citation. Please find the latest one. Because I saw one of your citation is 1982. So it's not valid. Your research, if you refer too long, you must find within 5 years at least. 80% of your total citation is references should be within 5 years. Okay, thank you. That's all from me.
Doctor.
Thank you, Nazifa. So I think that's all for now. Is the end of the session. I would like to thank every presenters and then all the audience for supporting your friends. So hopefully that you are able to finish your study on time. Okay? So thank you. So have a good day and Assalamualaikum.